Good afternoon. I'm Leigh Johnson. I'm going to present some of the work that we've been pursuing with Astragalus anserinus over the last couple of years. And this work prominently features the help of a couple of undergraduate students, Brenden, Thomson, and Martin Batalla. And I really appreciate their help. I also appreciate the help and financial support of the Bureau of Land Management and helping gathering some of the tissues and samples that we're using in our studies. So most of us have been in the field and experienced Astragalus, and we know that it can be a challenge to identify. Part of that is because keys are often written that require mature fruits to identify, while at the same time having mature flowers. And then when we're in the field, we often have plants that are either just in the fruiting stage or just in the flowering stage, trying to decide whether it's subcaulescent or acaulescent. Can sometimes be difficult, and a bunch of other difficulties with Astragalus that make it a taxonomic and identification challenge. So adding to the taxonomic challenge with Astragalus is that there's a large number of species. With 2,500 species, Astragalus is actually the largest genus of flowering plants. And here in the New World, we have about 500 species. These 500 species are the descendants of an ancestral polyploid event that had 4 sets of chromosomes, and then some of the chromosomes have been lost over time. That makes these aneuploid, which means as we look at the nuclear genome, most gene copies are actually at least duplicated. Many Astragalus have a perennial habit, and there's generally an association with the rate of DNA sequence evolution with the lifespan, so that things that have a longer lived lifespan have a little bit lower rate of DNA sequence evolution, and then that can impact our ability to reconstruct phylogeny with some of the commonly used regions for phylogenetic reconstruction. You compound that with the relatively rapid radiation in terms of large number of species that have specialized on special soil types. We have narrow distributions, and so lots of kind of specific niches that different Astragalus occur in. Some are widespread, but others are very narrowly endemic. And all these things add to the challenge of understanding relationships in Astragalus and understanding what's going on phylogenetically and taxonomically with this genus. Some of those things I mentioned, again, lead to phylogenetic challenges. Back in the turn of the century, studies were done with sampling with some of the common chloroplasts in the nuclear ribosomal cistron Astragalus, trying to look at phylogenetic relationships. And for us, there was a single species of interest, Astragalus persii included. But this is Neo-Astragalus. These are the New World species. And you can see that there's very low resolution across all of that diversity of 350 to 500 species that we have in the New World. A few years later, a study was done of Astragalus with some of those same genes and additional. There are only 2 New World Astragalus species sampled, and so that doesn't give us very much of a framework in terms of how to begin looking at Astragalus anserinus and how it's related to other Astragalus species. What we do have, though, is Barnaby in 1964 proposed a classification of Astragalus that's based on his study and his look at morphology, his intuition as to what characters were primitive and which ones were derived. And he recognized a number of sections in one of these sections is section Argophyli, and within that section of Argophyli is subsection Eriocarpi. And it's within subsection Eriocarpi that Astragalus persii, Astragalus anserinus, Astragalus utahensis occur. So this gives us a framework in terms of how to begin looking at Astragalus anserinus and addressing some of the questions that we had with this species. So Astragalus anserinus, or the Goose Creek milkvetch, has a pretty narrow distribution. It's known primarily from the Goose Creek drainage system, here where Utah meets with Idaho and Nevada. The populations span an area from about 20 miles long and about 4 miles wide. The species is typically found on tufaceous volcanic outcrops of the Salt Lake Formation, and there's a large number of species, or a large number of individuals prior to a fire that swept through the area. I think in 2007, about 60,000 plants were found inside this area, but it does have a rather narrow and restricted distribution. So although the main population center of Astragalus anserinus is along the Goose Creek drainage here, there is a population that was collected in 1999 over here in western Elko County, Nevada, that morphologically seems consistent with Astragalus anserinus. Rupert Barnaby, the expert at the time, identified these plants as Astragalus anserinus, and it's led to some question on whether or not these individuals really are the same as Astragalus anserinus that occur here in its really known distribution. NatureServe, if you look at the NatureServe Explorer, they actually discuss this, and they say a recent disjunct report from the YP Desert of northwestern Elko County, Nevada, was based on misidentified specimens of Astragalus pershii. This is interesting because I'm not too sure what the basis of that comment is in terms of where it's documented. If you look at plants, Astragalus anserinus from the Goose Creek drainage and Astragalus anserinus from the Owyhee Desert area of Elko, Nevada, you'll see that their fruits are very similar to each other, and they lack the dense tomatose pubescence that we find on fruits of Astragalus pershii. So the question is this really a misidentified Astragalus pershii? If it is, it's actually quite different in its fruit morphology compared to Astragalus pershii, regardless of which variety it is that we're looking at. So our study was really to answer a pretty simple question. Is Astragalus anserinus from the Goose Creek drainage conspecific with Astragalus anserinus from the Owyhee Desert? And we were able to answer this with the help of the BLM in terms of obtaining some leaf tissue. And from that leaf tissue, we were able to do some initial chloroplast sequencing the entire chloroplast genome. And then we were able to look specifically for highly variable regions of that genome between samples to design some specific primers that would give us greater phylogenetic resolution than the regions that were used in the earlier studies that didn't show very much differentiation among taxa at all. So our plant sampling included 8 samples from the Goose Creek area of Astragalus anserinus and 4 samples from the Owyhee area. We included at least 2 samples of each of the subspecies of Astragalus pershii. We included other species from section Eriocarpi with at least 1 sample of each one. There are 3 species that we do not have in our herbarium, not been able to sample until this coming field season. We plan on obtaining fresh tissue for these species and can add them into our analysis. And then we looked at least 1 sample of each of the other subsections of section Argophyllae, trying to do our best just to provide a strong phylogenetic context for the results that we have in comparing the samples of Astragalus anserinus. So Parsimony Analyses, we've covered a number of most parsimonious trees, but they're really pretty similar to each other and differ only in these really short branches here. Our outgroups are old world species of Astragalus, and we included Astragalus oophorus, which is supposed to be outside of section Argophyllae. But what we found is that Astragalus perii from section Argophyllae is actually sister to the rest of our samples, including Astragalus oophorus, which makes section Argophyllae not monophyletic. This is perhaps not too surprising since Barnaby's classification is based largely on intuition, but you have to start somewhere, and in the absence of phylogeny, we use previous classifications as a proxy for that phylogeny. Taking a step further, subsection Eriocarpi, of which Astragalus anserinus belongs, is not monophyletic. All of the taxa that are highlighted in the teal color belong to section Eriocarpi, and you can see that embedded among those are various species from other subsections of section Argiophili. And more to the point, with the species we're interested in, Astragalus anserinus actually occurs in two really widely separated clades, more closely related to a number of other taxa from section Argiophili and subsection Eriocarp. So more specifically, Astragalus anserinus from the Goose Creek area, the type locality, actually shows relationships with Astragalus pershii variety tinctus, which occurs over in the Waihi Desert of Nevada, and then other species from section Argiophili. The anserinus from the Waihi Desert actually is embedded in a clade that includes most of the other varieties of Astragalus pershii, which is interesting in terms of being potentially a misidentified Astragalus pershii. But it is very well supported as its own group and much more differentiated from most of the other Astragalus pershii individuals that were sampled. So just rethinking the relationships of the Waihi individuals, if they really are a variety of Astragalus pershii, it causes us to rethink actually what is Astragalus pershii, because these individuals are very unlike Astragalus pershii in their fruits and some other characteristics. And Astragalus pershii itself is coming out in multiple places in our phylogenetic tree. So this is perhaps not too surprising if we look at this quote from Isley. Astragalus pershii is an intricate, multiracial complex of confluent varieties, most of which are closely related to other species. The claim of some of these, as AU taensis and A inflexus, to specific status may be little more than that of several of the taxa here assigned to Astragalus pershii. So to conclude, Astragalus anserinus from the Ohi Desert is not conspecific with Astragalus anserinus from the primary population center in the Goose Creek drainage. However, the Ohi population is more than just a misidentified pershii. It's morphologically distinct from any named variety of pershii, and further work is needed to characterize it morphologically more fully. Our current efforts to add nuclear data via amplicon sequencing will provide further insights into species phylogeny in this group. And there's still considerable work that remains to understand the relationships among taxa of Astragalus section Argiophili, including the Goose Creek milkvetch. Thank you.